Okay, so welcome back to this next video in which we are discussing high-density lipoprotein and the reverse cholesterol transport process. Okay, right, so I'd like to begin this video with a small correction to something that I said in the previous video. So in the previous video, what I told you is that in cells which are overloaded with cholesterol, you get a rise in the level of these oxysterol compounds. These oxysterol compounds then bind to the liver X receptors, which are within these liver X receptor, retinoid X receptor heterodimers, okay? And that when the oxysterol binds to the liver X receptor with the 9-cis retinoic acid, this heterodimer complex with the two ligands attached then binds to a coactivator, and that increases the expression of ABCA and ABCG1, which then ABCA1 and ABCG1, which then trigger increased cholesterol and lecithin efflux from these cholesterol overloaded cells. Okay, now that's perfectly correct. However, I implied that you required both oxysterol and the 9-cis retinoic acid in order to activate this liver X receptor, retinoid X receptor heterodimer. In actual fact, one of those two ligands is enough to activate the heterodimer and get a coactivator binding to the heterodimer, and then activate the transcription of the genes. Okay, if you get both of them binding together, they do actually synergize one another, and the activation of expression of the genes is greater than what you would expect just from the two adding together. Okay, but you don't need both of them, so the oxysterol on its own without 9-cis retinoic acid is enough to activate this liver X receptor, retinoid X receptor heterodimer. Okay, right, so that then leads to the expression of ABCA1 and ABCG1 going up on the surface of our cholesterol overloaded cell, and now the cholesterol overloaded cell is going to efflux cholesterol and lecithin molecules onto these small discoidal HDL molecules which have come from the liver and from enterocytes. Okay, so what happens is more and more cholesterol and lecithin go into these small discoidal HDL molecules. Now, that's not good. Well, that's not useless on its own, because how are we going to turn this into a spherical HDL when we haven't got anything to put in the core, basically, because both the lecithin molecules and the cholesterol molecules will go in the phospholipid monolayer, okay? But we have this enzyme, lecithin cholesterol acyl transferase, which will transfer a long-chain carboxylic acid, which is esterified to the second alcohol group of the glycerol molecule within a lecithin molecule, onto the alcohol group of a cholesterol molecule, creating us a cholesterol ester and a lysophosphatidylcholine molecule. The lysophosphatidylcholine, also called lysolecithin, will remain in the phospholipid monolayer, whilst the cholesterol ester can go into the lipid core. Okay, so now what's going to happen is these HDL molecules, which are now spherical HDL molecules, are going to go back to the liver, okay, or they will also go to steroidogenic cells, which use cholesterol as the precursor to the steroid hormones. So, for instance, cells of the adrenal glands, cells of the testes, and cells of the ovaries. Okay, so this is now called a spherical HDL molecule. Okay, however, there's another way that these HDL molecules can empty their cholesterol ester core. They can also transfer the cholesterol esters to LDL particles, and for this we need to now discuss this second type of lipoprotein, LDL. Okay, so LDL is another type of lipoprotein which delivers cholesterol esters. So the main thing at the hydrophobic core of an LDL lipoprotein is going to be cholesterol esters. So LDL stands for low density lipoprotein. Okay, and the density in the name refers to how dense the protein is within the lipid. So remember, when we saw HDL, we saw that you had a lot of lipid molecules and then some protein molecules. And the ratio of the protein to the lipid determines how dense the protein is within the lipid. So high density lipoproteins have a relatively high amount of protein for the amount of lipid they have, whereas low-density lipoproteins have much lower levels of protein for the amounts of lipids they have. Okay, so it's a very similar structure to HDL particles. Okay, so they are slightly bigger than HDL particles. They generally have a diameter between 20 and 25 nanometers. So they are slightly bigger, but the structure is effectively the same. They have a phospholipid monolayer that makes up the outer surface of the sphere, and then they have a hydrophobic core. Now, the special thing about LDL particles is that they only have a single apolipoprotein within their phospholipid monolayer. Okay, and this apolipoprotein is always a set apolipoprotein. So absolutely always, an apolipoprotein B100 will be in the phospholipid monolayer of an LDL particle, and this will be the only apolipoprotein in the phospholipid monolayer. So you have one apolipoprotein B100, and that is it as far as the apolipoproteins are concerned. Okay, so you don't have any more within the LDL molecule. Now, apolipoprotein B100 is a giant of a protein. It's easily the biggest apolipoprotein, and it's a very, very big protein, close to 5,000 amino acids long, so it's a big, big giant of a protein. So it dominates the phospholipid monolayer of this LDL particle. Okay, the rest of the phospholipid monolayer then is made up of phospholipids, the main example again being lecithin. Okay, and you will again have free cholesterol molecules dotted in amongst the lecithin molecules which make up the phospholipid monolayer. Okay, so that's the outer shell of these low-density lipoproteins. Then in the hydrophobic core of the low-density lipoprotein, what you have is a lot of cholesterol esters, just like HDL. Okay, so you'll have many, many esters of cholesterol. So I'll try and show these like so. I might draw a little blue square here, and then put a long chain carboxylic acid sticking off like so. Okay, so that can denote a cholesterol ester, and then we'll have the free cholesterol molecules denoted just in blue here in the phospholipid monolayer. Okay, now the thing which you do have a tiny little bit of in low-density lipoprotein particles, which you don't have in high-density lipoprotein particles, at least not until they've interacted with LDL particles, is triacylglycerols. Okay, so this is time to discuss another type of lipid molecule. Okay, so triacylglycerols are often abbreviated to TAGs. Okay, so the T is for tri, the A is for acyl, and then the G is for glycerol. Okay, so TAGs for short. Now they have another name. They're also often called triglycerides. Okay, so let's draw a little picture or a cartoon for triacylglycerols, like we did for the phosphoglycerolipids. Okay, so once again, the backbone structure for them is a glycerol molecule or a propane 1,2,3 triol molecule. Okay, and this time what we're going to do is we're going to esterify onto the free alcohol groups of this glycerol molecule long chain carboxylic acids. So we're going to put long chain carboxylic acids on all three of the alcohol groups. Now these long chain carboxylic acids do not need to be the same long chain carboxylic acid. Okay, so you can have stearic acid here, palmitic acid here, linoleic acid here, whatever you want basically. So there are many different triacylglycerol molecules. Now, these are extremely hydrophobic. Most of their structure is neutral, 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 so they do not interact well with water at all. So when they're being transported around the blood, they need to be in lipoproteins, and they need to be in the hydrophobic core of lipoproteins. And you have a few triacylglycerols within low-density lipoproteins. It's not a massive number, okay? Not compared to something like a chylomicron or a very low-density lipoprotein, which we're not going to talk about in this video, but they do have triacylglycerols. And now what's going to happen basically is the low-density lipoprotein is going to take cholesterol esters from the high-density lipoprotein, okay, and in exchange it will give the high-density lipoprotein a triacylglycerol. Okay, so here we're drawing the high-density lipoprotein here. And what will happen is the high-density lipoprotein will come into contact with the low-density lipoprotein here. Now, this is going to involve another protein which is on the surface of high-density lipoproteins, which we haven't talked about before. Okay, so let's just remind ourselves of the structure of a high-density lipoprotein. Okay, so we've got our apolipoprotein A1 being the main apolipoprotein within the phospholipid monolayer of our high-density lipoprotein. Okay, we've then got lecithin cholesterol acyl transferase enzymes also embedded within the phospholipid monolayer. And of course, a major difference between HDL and LDL is that you can have multiple apolipoproteins as well, and there's vast scope for different apolipoproteins to be in the phospholipid monolayer of a high-density lipoprotein particle. Now, the important protein that I haven't mentioned before is now going to be involved in this swap of a cholesterol ester from the core of the high-density lipoprotein to the core of the low-density lipoprotein in exchange for a triacylglycerol coming from the core of the low-density lipoprotein into the core of the high-density lipoprotein. Okay, now this protein, and I'm not going to colour it in blue because then it will get confused for the cholesterol molecules. Okay, I'll colour it in yellow. This protein is known as cholesterol ester transfer protein. Okay, so in yellow there, this is the cholesterol ester transfer protein. And for short, the cholesterol ester transfer protein is often abbreviated to its initials. Okay, so C for cholesterol, E for ester, T for transfer, and then P for protein, so the CETP for short. And basically it's going to move a cholesterol ester into the LDL molecule in exchange for a triacylglycerol coming into the core of the HDL molecule. Okay, so high-density lipoproteins can therefore give away their cholesterol esters to LDL molecules in exchange for triacylglycerols which come from the LDL molecules. Okay, right, so the HDL particles have got these cholesterol esters from cells which are overloaded with cholesterol. The cells which were overloaded with cholesterol, remember, gave their HDL particles cholesterol and lecithin, and the LCAT then produced cholesterol esters from those two substrates. Okay, and now the HDL particles can either go and give this cholesterol directly to the liver, or they can give it to low-density lipoproteins, which can also give it to the liver. So what we now want to look at is how both HDL particles and LDL particles deliver their cholesterol esters to the liver and also to steroidogenic cells in the adrenal glands and also the testes and ovaries. Okay, so let's now talk about this. So, right, let's start by discussing the two major cell types that we're going to deliver the cholesterol to. Okay, so we have liver cells firstly. So here's a picture of the liver, and we'll have our hepatocyte represented here. Okay, and HDL particles and LDL particles are now going to deliver their cholesterol ester content to the hepatocyte. Okay, now they'll do this through very different mechanisms, as we'll see, but the overall result of this is that the cholesterol that the hepatocytes receive is either going to be excreted into the bile directly as a free cholesterol, okay, or it's going to be converted into a bile acid, and then the bile acid will be put into the bile. Okay, so we're going to use cholesterol to make bile. So bile contains both free cholesterol molecules and also bile acids, and it's important in the absorption of lipid molecules from the small intestine. Okay, right. So hepatocytes is one of the targets for HDL and LDL to deliver their cholesterol esters to. Another target is the steroidogenic cells. Okay, right. So let's firstly discuss the adrenal gland. Okay, so the steroidogenic cells. So we'll start with the adrenal glands then. Okay, so you have two adrenal glands that sit atop the kidneys. So you have a left adrenal gland which sits atop the left kidney, and you also have a right adrenal gland which sits atop the right kidney. Okay, and for this reason they're also occasionally called the suprarenal glands. Okay, because they're above the kidney. Okay, so suprarenal glands or adrenal glands. Okay, so basically adrenal glands can be divided up into two main portions. Okay, so there is the medulla of the adrenal gland, which is the middle portion, and I'll colour this in orange here. So this orange portion here, this represents the medulla, and then there is the outer portion, which I'll colour in purple here, which is the cortex of the adrenal gland. Okay, now the adrenal medulla is going to be involved in the synthesis of adrenaline, okay, which is then secreted into the blood in times of stress. Whereas the adrenal cortex is going to be involved in the synthesis of all sorts of steroid hormones. Okay, so this is the part we're interested in, the adrenal cortex, rather than the adrenal medulla. Okay, now the cortex is further divided into three separate layers. Okay, so let me show you these. So we have zona glomerulosa is the most outer layer. Okay, so it's the most peripheral layer, the layer that's you'd reach to first. If you were drilling in from the outside, you'd reach the zona glomerulosa first. Okay, and this layer is responsible for the production of the steroid hormone aldosterone, which affects kidney function. Okay, so it makes the kidney reabsorb more water, basically. And it's got an important role in maintaining body fluid volumes, and it's involved in the renin-angiotensin-aldosterone system. Okay, the second layer that's underneath zona glomerulosa is zona fasciculata. Okay, and this is involved in the synthesis of glucocorticoids, the main example being cortisol. Okay, which is involved in blood glucose regulation, hence why it's called a glucocorticoid, but also it's involved in the more general response to stress, basically. And it's a very difficult hormone. It does many different things, and it's quite difficult to pinpoint what its actual function is. With aldosterone, it's very obvious what its major function is, and we can sort of ignore its more minor functions, whereas cholesterol, it has loads of kind of mediocre important functions, and no sort of clear function that dominates over all the others. Okay, and then underneath that, we then have zona reticularis. Okay, and this layer produces sex hormones, okay, so androgens, estrogens, things like that. Okay, so all of them synthesize steroids, and the starting point for the synthesis of steroids is cholesterol. So they need a supply of cholesterol to make these hormones. So LDL molecules and HDL molecules, which are full of cholesterol esters, can bring them to these cells of the adrenal cortex, basically. Okay, in addition, you also have the testes and the ovaries, which we can summarize just as gonads, basically. So gonad means either a testis or an ovary. Okay, and basically these also secrete sex hormones. Okay, so the testes secrete large amounts of androgens, the ovaries secrete estrogens and progestogens. Okay, so again, the starting point for the synthesis of those steroid hormones is cholesterol, so they also will uptake these HDL and LDL particles to deliver cholesterol esters to the cell, and so that those cholesterol esters can be used in the production of the steroid. Okay, right, so we'll call it there for this video, and in the next video what we'll look at is the pathways by which cells can take cholesterol esters from HDL particles and from LDL particles, and these mechanisms differ hugely. LDL is going to be endocytosed completely into the cell, and it's going to be completely broken down, whereas HDL is just going to interact with a receptor in a more normal way, and the receptor will just extract the cholesterol ester from it, and it won't be broken down in the same way that LDL is broken down. Okay, right, and we'll focus on these pathways for harnessing the cholesterol within HDL and LDL particles within liver cells rather than the steroidogenic cells.